Assalamu alaikum my dear students. So hematology में या blood में we are actually done with the composition and functions of blood, and we have also discussed about the composition of plasma. We are also done with the plasma proteins. Now we are going to talk about the formed elements, and we have already talked about that formed elements of blood जो है इसमें there are three types of cells. We have red blood cells, we have white blood cells, and we have platelets. Now we have to talk about the all these types of blood cells, and we will talk about the red blood cells or erythrocytes first. And in this lecture today, we are just going to have an introduction to red blood cells, and then we will focus on the morphology of red blood cells. Let us get started. So when we talk about the red blood cells actually जो है, they are also called as erythrocytes. And erythrocytes it is made up of two words actually, in which erythro, let me write it here, erythro actually means red and cytes means cells. So simply they are actually the red cells, right? And these red cells we also call them as red blood cells. And these red blood cells are actually among all the three types of formed elements or three types of blood cells. These red blood cells are the most abundant type of cells which are present in the blood. And these red blood cells erythros means red and cyte means cells. These cells are red in color because of the presence of a coloring pigment inside, and that coloring pigment is called as hemoglobin. So these red blood cells actually contain hemoglobin in their cytoplasm, and that hemoglobin, because of the presence of that hemoglobin inside, these cells appear red in color. So we simply call them as red blood cells or the red cells or erythrocytes. Now, I have told you that these are the most abundant type of cells. If you remember, in hematocrit, I have told you, जब हमने हेमाटोक्रिट ट्यूब में जो है फिर सेंट्रीफ्यूज में डाल के जो है एक्चुअली हमने पैक्ड सेल वॉल्यूम जो है देखा था, तो पैक्ड सेल वॉल्यूम में 45% ऑफ दैट गिवन अमाउंट ऑफ ब्लड वाज रेड ब्लड सेल्स. So we can understand कि these red blood cells जो है, when we compare these red blood cells to the platelets and the white blood cells, these red blood cells are the most abundant type of cells. And when we talk about the normal count, वो भी हमने वहां पे बता दिया था in the hematocrit. When we talk about the normal count of red blood cells, normal count is actually, in some books it is 5 to 6 million per cubic millimeter. In some books it is 4 to 6 per cubic millimeter. 4 to 6 millions. So let me write it here. It is 4 to 6 million per cubic millimeter. So in 1 millimeter cube there are 4 to 6 or 5 to 6 million red blood cells. But in females, the RBC count, the red blood cell count, is slightly on the lower side. So average जो count होता है in females, average count, that is 4.5 million in females. So it is 4.5 million per cubic millimeter, and in the males it is 5. The average count that is 5 million per cubic millimeter. So 5 million in males. So what did I tell you is that RBCs they are actually non-nucleated cells. We are going to talk about that, and they are non-nucleated cells, and among all the formed elements, these red blood cells are the most abundant type of cell among all the types of blood cells. And these red blood cells they are called as erythrocytes. Erythro means red and cytes means cells, so they are red cells, and they are red in color because of the coloring pigment present in the cytoplasm, that is called as hemoglobin. Now let us talk about the morphology of red blood cells. And morphology of red blood cells में we are going to talk about the normal shape of RBCs, normal size of RBCs, and normal structure of RBCs. So that is actually we are going to talk about in the morphology of red blood cells. So let us talk about the normal shape first. Now, when we talk about the normal shape, actually जो ये red blood cells होती है, they are disc shaped, और we call it as जो है ये RBCs जो है they are biconcave in shape, biconcave shaped. Now, look here in this diagram. For example, we have this RBC, and this RBC, the shape of the RBC actually जो है इसके shape पे there are two concave surfaces. There are जो है, and this concavity जो है ये दोनों sides में है, and this concavity is in the center. For example, to understand this, for example, I am taking a cross section in this RBC. I am cutting this RBC like this, and then I am looking at the cross section. Look here. Now, in the shape of the RBCs, we can see here that there is the concavity in this side, there is the concavity in this side. So we call it as the biconcave shape of RBCs. So, and we also call it as dumbbell shaped. You can see here it is जो है जब हम इसका cross section देखते हैं तो we find this shape as the dumbbell shaped, and this dumbbell shape is actually thin in the center and it is thicker in the peripheries. We are going to talk about the thickness in the normal size of RBCs, but here we are talking about this normal shape of RBCs. So the normal shape of RBCs is actually they are the disc shaped, or they are the biconcave shaped, and there is concavity on both the sides, and that concavity is actually in the center. Right? So we are going to find the RBCs जो है, they are actually thinner in the center and they are much thicker in the peripheries. We are going to talk about that. Now, this biconcave shape जो है, it has got some benefits there in the RBCs. Why there is this biconcave shape in the RBCs? Now for that, let me tell you कि RBCs जो होती है, RBCs का जो important function है, एक्चुअली हमारे ब्लड में जो आरबीसीज का जो सबसे इंपॉर्टेंट फंक्शन है, दैट इज एक्चुअली द ट्रांसपोर्ट ऑफ गैसेस, दैट इज द ट्रांसपोर्ट ऑफ ऑक्सीजन एंड ट्रांसपोर्ट ऑफ कार्बन डाइऑक्साइड. नाउ in order to जो है transport this oxygen and जो है बिकॉज ऑक्सीजन जो है हमें लंग्स से सारे बॉडी टिश्यूज तक पहुंचाना होता है, और कार्बन डाइऑक्साइड हमें बॉडी टिश्यूज से वापस लंग्स में जो है पहुंचाना होता है. Right? So that is the function of RBCs. Now, why there is biconcave shape in the RBCs? The first thing is that this biconcave shape actually it increases the surface area of the RBCs. It increases the surface area of the RBCs. So what is the surface area? We are going to talk about that in the normal size. But here you have to remember that this biconcave shape is actually important to increase the surface area of the RBCs. And when there is increased surface area in the RBCs, that means there is enough space for the diffusion of these gases, for the diffusion of जो है oxygen, for the diffusion of carbon dioxide. In order to deliver the oxygen to all the tissues, there should be enough space in the RBCs. In order to receive this carbon dioxide from the tissues and deliver it to the lungs, there should be enough जो है surface area. अब surface area को बढ़ाने के लिए this biconcave shape is very important. So the first benefit of this RBC जो है biconcave shape of the RBCs is it increases the surface area for the diffusion of the gases. And the second जो है biconcave shape का जो second benefit है, and that benefit is actually this biconcave shape helps the RBCs to squeeze through the smaller capillaries in our body. Squeeze through smaller capillaries. We have small capillaries, और उन capillaries का जो diameter होता है, that diameter is very very small, और उन capillaries के through जो है इन RBCs को जो है pass होना पड़ता है, squeeze होना पड़ता है, तो उसके लिए जो है this biconcave shape is very important. If there is no biconcave shape, that means the RBCs will be spherical and it will be very difficult जो है for these RBCs to pass through these smaller capillaries. So the first benefit is it increases the surface area, and second benefit of the biconcave shape is that it helps the RBCs to squeeze through the minute capillaries in our body. So normal shape में these the normal shape of RBCs is actually, it is the disc shaped or the biconcave shaped cells, and when we take the cross section we find dumbbell shape, and that dumbbell shape means in the center it is thinner and in the peripheries it is thicker. So that is the shape of RBCs, and this biconcave shape is actually very important for these RBCs to increase the surface area and to pass through the smaller capillaries in our body. Now let us talk about the normal size. Now normal size में हम actually चार चीजें जो है वो discuss करने वाले हैं. The first thing is the diameter of these RBCs. The first thing we are going to talk about is the diameter. So diameter of these RBCs is actually, when we talk about the average diameter, that means this is the diameter here. So from this end to this end, like this is the diameter, and the diameter of these RBCs, the average diameter is actually 7.2 microns. 7.2 microns is the average diameter of these RBCs. You have to remember this. The second thing in the normal size we are going to talk about is thickness. Thickness of the RBCs. And thickness में मैंने आपको अभी कहा that when we talk about the thickness of RBCs, you can find it here कि thickness जो है RBCs में it varies from the peripheral area to the central area, right? So central area of the RBCs is actually thinner as compared to the peripheral area of the RBCs. So there is the thickness actually varies from the central to the peripheral area of the RBCs. And in center, the thickness is actually center में जो thickness है that is approximately 1 microns, and जो peripheries है peripheral area में जो thickness है that is approximately 2.2 microns, right? So the diameter is, average diameter is 7.2 microns, and the second thing is thickness, and thickness में जो है thickness varies from center to periphery. Center is thinner as compared to the periphery. Center में जो thickness है that is 1 micron, and in the peripheral area it is 2.2 microns. Now the third thing we have to talk about in the normal size, and that is surface area. Surface area. And surface area, मैंने अभी आपको कहा that biconcave shape actually helps the RBCs to increase the surface area. There should be enough area for the diffusion of जो है these oxygen and carbon dioxide, and the surface area, the normal surface area of the red blood cells is approximately 120 square microns. So it is approximately 120 square microns. And the last thing we are going to talk about in the normal size, and that is the volume of RBCs. Volume of RBCs, and the volume of RBCs is actually going to be 85 to 90 cubic microns. So normal size mein we have seen that we have to cover 4 things. First thing is diameter. Average diameter is 7.2 microns. In some books you may find it as 7.5 microns. That is all right. So second thing is thickness, and thickness in the center is 1 micron, and in the periphery it is 2.2 microns, and then the surface area is 120 square microns, and the volume is 85 to 90 cubic microns. Right? Now let us talk about the normal structure of the RBCs. Now normal structure mein I have already told you that there will be these RBCs actually जो है they are non-nucleated cells. The first thing you have to remember in the normal structure is that these are non-nucleated cells. Let me use this red color only. So these are non-nucleated cells. And mammals में जो है the only जो है where this RBCs जो है मतलब न्यूक्लिएटेड होती है, दैट इज कैमल में RBCs जो है दे आर न्यूक्लिएटेड. इन ऑल द अदर मैमल्स जो है ये RBCs जो है दे आर नॉन न्यूक्लिएटेड. And non-nucleated cells are there. So if there is no nucleus present, definitely जो है न्यूक्लियस में जो है हमारा DNA प्रेजेंट होता है. So RBCs में DNA भी प्रेजेंट नहीं होता है. राइट, इन हमारे आरबीसीज में डीएनए प्रेजेंट नहीं होता है. पहले होता है न्यूक्लियस, लेकिन ड्यूरिंग द डिफरेंट स्टेजेस ऑफ आरबीसी फॉर्मेशन, जब हमारे पास मैच्योर आरबीसी जो है फॉर्म होती है, तो उनमें एक्चुअली न्यूक्लियस जो है वो प्रेजेंट नहीं होता है. सो न्यूक्लियस जो है वो एक्चुअली इजेक्ट आउट होता है. वो हम देखेंगे एरिथ्रोपोइसिस प्रोसेस में. फॉर्मेशन ऑफ आरबीसीज जो है हमारा एक इंपॉर्टेंट टॉपिक है, वेरी इंपॉर्टेंट टॉपिक, फॉर्मेशन ऑफ आरबीसीज दैट इज एरिथ्रोपोइसिस. उसमें हम देखेंगे कि किस स्टेज तक जो है वो न्यूक्लियस प्रेजेंट होता है और फिर किस स्टेज में जो है न्यूक्लियस डिसअपियर होता है. एंड नॉट ओनली न्यूक्लियस इज एब्सेंट देयर इन द आरबीसीज, अदर सेल ऑर्गेनेलीज जो है, अदर सेल ऑर्गेनेलीज like mitochondria, like ribosomes, like जो है the Golgi apparatus, all the other cell organelles जो है important cell organelles, they are also absent. Like most importantly we will see mitochondria. So mitochondria जो है वो इन cells में absent होता है. Now the thing is that mitochondria is actually the powerhouse of the cell. So Krebs cycle जो होता है वो हमारे mitochondria में ही होता है, जो ATP formation का जो है एक source होता है cells के लिए. तो अगर mitochondria इन cells में absent है, तो इन cells को जो है RBCs को ATP जो है source कौन सा जो है metabolic pathway होता है या जो है metabolic process होता है, that is actually glycolysis. So these RBCs जो है वो इनका जो एनर्जी सोर्स होता है, एनर्जी सोर्स दैट इज ग्लाइकोलाइसिस. और ग्लाइकोलाइसिस के लिए जो है हमारे पास माइटोकॉन्ड्रिया की जरूरत नहीं होती है. तो ग्लाइकोलाइसिस से ही आरबीसीज को जो है एनर्जी हासिल होती है. और इसके बाद जो ग्लूकोज सेल्स के अंदर जाता है, तो यूजुअली हम क्या देखते हैं, तो ग्लूकोज जब मतलब ग्लूकोज अपटेक सेल्स के द्वारा जो है वो जब होता है, तो ग्लूकोज सेल्स के अंदर जाने के लिए वी हैव डिफरेंट ट्रांसपोर्टर्स, वी कॉल देम एज ग्लूकोज ट्रांसपोर्टर्स. एंड सम ग्लूकोज ट्रांसपोर्टर्स आर इंसुलिन डिपेंडेंट ग्लूकोज ट्रांसपोर्टर्स, एंड सम ग्लूकोज ट्रांसपोर्टर्स ग्लूकोज डिपेंडेंट के लिए, फॉर एग्जांपल GLUT4 वी हैव ग्लूकोज ट्रांसपोर्टर टाइप 4. That is insulin dependent glucose transporter. That means cells glucose cell के अंदर जाने के लिए through GLUT4 और that insulin dependent, we need insulin there. जब insulin cell को stimulate करेगा तभी जो है वो ग्लड ट्रांसपोर्टर जो है या ग्लूकोज ट्रांसपोर्टर जो है एक्टिवेट हो जाएगा, तभी वो ग्लूकोज को सेल के अंदर ले पाएगा. लेकिन जो आरबीसीज है, आरबीसीज के ऊपर सेल मतलब आरबीसीज मेम्ब्रेन पे देयर आर नो इंसुलिन रिसेप्टर्स, नो इंसुलिन रिसेप्टर्स. सो इंसुलिन रिसेप्टर्स अगर नहीं है, तो इंसुलिन रिसेप्टर्स की एब्सेंस में व्हाट डज इट मीन इज कि जो ग्लूकोज इन आरबीसीज के अंदर जाता है दैट गोज वाया दी इंसुलिन इंडिपेंडेंट रिसेप्टर्स, सॉरी इंसुलिन इंडिपेंडेंट ग्लूकोज ट्रांसपोर्टर. सो जो ग्लूकोज ट्रांसपोर्टर आरबीसी के मेम्ब्रेन पे होता है, फॉर एग्जांपल हियर इज द आरबीसी मेम्ब्रेन फॉर एग्जांपल, एंड हियर इज द ग्लूकोज ट्रांसपोर्टर, एंड दिस ग्लूकोज ट्रांसपोर्टर जो है, फॉर एग्जाम्पल हियर इज द ग्लूकोज ट्रांसपोर्टर. अब किसी सेल में, फॉर एग्जाम्पल दी मसल्स, फॉर एग्जाम्पल दी एडिपोज टिश्यूज, उनमें जो ग्लूकोज ट्रांसपोर्टर होता है दैट इज इंसुलिन डिपेंडेंट. जब इंसुलिन आएगा, फॉर एग्जाम्पल हियर इज दी मसल सेल, जब इंसुलिन यहां पे होगा तो ये इंसुलिन जब यहां पे सेल पे एक्ट करेगा, तभी ये सेल जो है ग्लूकोज को अंदर ले पाएगा. राइट, तभी ये सेल जो है ग्लूकोज को अंदर ले पाएगा. लेकिन इस सेल के अंदर जो है ग्लूकोज जाने के लिए वी डू नॉट नीड इंसुलिन देयर. Right, we do not need insulin, so we call this glucose transporter as insulin independent glucose transporter, and it is most importantly GLUT type 1. So we have glucose transporter type 1, which is actually insulin independent glucose transporter, right? So no insulin receptors, that means जो glucose transporter वहां पे है, we have the GLUT1 transporter there, which is insulin independent transporter. Insulin independent. Now one more important thing is that I have told you कि RBCs जो है, they are biconcave shaped. Now to attain this biconcave shape, there is a special protein in the RBCs, in the cytoskeleton of RBCs. जो cytoskeleton of RBCs होता है, वहां पे दो important proteins होते हैं. One is when we talk about the cytoskeleton of RBCs, cytoskeleton, we have there actin and spectrin. एक्टिन तो हम यूजुअली देखते हैं इन द डिफरेंट सेल्स, वी हैव साइटोस्केलेटन में एक्टिन, एंड इन द आरबीसीज वी हैव अ स्पेशल प्रोटीन देयर व्हिच इज कॉल्ड एज स्पेक्ट्रिन. एंड दिस स्पेक्ट्रिन, फॉर एग्जांपल, हियर इज द स्पेक्ट्रिन प्रोटीन. फॉर एग्जांपल, here is the spectrin protein which is present in the cytoskeleton of these RBCs, and suppose here we have the membrane proteins. Now this spectrin protein is actually connected with the transmembrane proteins by another protein. Suppose here is that protein which actually connects the spectrin with the membrane proteins, and that protein is called as ankyrin. One more time, we have in the cytoskeleton of RBCs, we have actin and we have spectrin protein. And this spectrin protein is connected with the membrane proteins. So membrane proteins or transmembrane proteins, the proteins which are present in the cell membrane of the RBCs. Now cell membrane वाले proteins spectrin के साथ connected होते हैं by another protein, and that protein is called as ankyrin. And you have to remember that to attain this biconcave shape, it is actually जो ये biconcave shape RBCs में होती है, it is because of the protein which is called as spectrin. So spectrin जो है, यही प्रोटीन जो है एक्चुअली सेल्स को बाइकनकेव शेप अटेन करने में हेल्प करता है. Right. Now suppose we have this spectrin protein absent in the RBCs. Suppose the spectrin protein is absent in the RBCs, and or the spectrin protein जो है ये deformed है या non-functional है ये spectrin protein. So that means RBCs जो मतलब biconcave shape attain करते हैं, तो they will not be able to attain a biconcave shape. तो वो एक hereditary disorder जो है वहां पे हो जाएगा अगर biconcave shape नहीं है RBCs में, and that hereditary disorder there, which is actually because of the absence of spectrin protein, that is called as hereditary spherocytosis. Hereditary spherocytosis. Hereditary spherocytosis. Hereditary spherocytosis जो है when the spectrin protein will actually be absent in the RBCs. So, अगर स्पेक्ट्रिन प्रोटीन हमारे आरबीसीज को बाइकनकेव शेप अटेन करने में हेल्प करता है, एंड वी हैव ऑलरेडी सीन हाउ इंपॉर्टेंट दिस बाइकनकेव शेप इज. हमारे सर्कुलेशन में जो आरबीसीज है ये एक्चुअली जो है आराम से पास हो सकते हैं थ्रू स्मॉलर कैपिलरीज बिकॉज ऑफ द बाइकनकेव शेप. If there is no spectrin protein which helps the RBCs to attain the biconcave shape, so that means RBCs will not be able to attain the biconcave shape. And if they are not able to attain the biconcave shape, the RBCs will be spherical in nature, right? In shape, the RBCs will be spherical, and the spherical structure of RBCs जो है, it will be very fragile. जो smaller capillaries के through जब pass हो जाएगा, तो it cannot pass easily. So there will be destruction of these RBCs, which will lead to hemolytic anemia, and that condition is called as स्फेरोसाइटोसिस. सो स्फेरोसाइटोसिस जो है इसमें सेल्स का डिस्ट्रक्शन होता है, हीमोलाइसिस होता है. हीमोलाइसिस मतलब ब्रेक डाउन ऑफ दीज आरबीसीज, और वो ब्रेक डाउन इसलिए होगा क्योंकि आरबीसीज में बाइकनकेव शेप नहीं होगा, और बाइकनकेव शेप इसलिए नहीं होगा क्योंकि इन आरबीसीज में जो है स्पेक्ट्रिन प्रोटीन प्रेजेंट नहीं होता है. So in normal structure we have seen they are non-nucleated cells, so there is no DNA present because no nucleus is present there, and other cell organelles are also absent. And जो ये nucleus नहीं होता है, जो है other cell organelles नहीं होते हैं in the mature RBC, it is mainly because कि RBCs के अंदर जो है enough space हो साइटोप्लाज्म में to accommodate the hemoglobin, और जब ज्यादा hemoglobin हमारे RBCs में present होता है तो ज्यादा जो है ऑक्सीजन हम सप्लाई कर सकते हैं to all the tissues of our body. So that is the main reason of this nucleus, absence of nucleus and other cell organelles in the RBCs. So we have seen other cell organelles are also absent, and most importantly the mitochondria is absent. And if mitochondria is absent, so there is no Krebs cycle. So जो energy source है RBCs के लिए, that is going to be glycolysis. और ग्लूकोज को अंदर लेने के लिए जो वहां पे ग्लूकोज ट्रांसपोर्टर होता है, दैट इज इंसुलिन इंडिपेंडेंट ग्लूकोज ट्रांसपोर्टर, बिकॉज आरबीसी मेम्ब्रेन पे इंसुलिन रिसेप्टर्स प्रेजेंट नहीं होते हैं. So insulin अगर receptors present नहीं है तो that means there should not be insulin dependent receptors, glucose transporters present there. So there are insulin independent glucose transporters present. So उसके बाद जो है cytoskeleton में there are two important proteins, actin and spectrin. और spectrin जो है ये membrane proteins के साथ connected होता है by N-chirin, और spectrin actually helps the RBCs to attain the biconcave shape. And if there is absence of this spectrin protein in the RBCs, so that will lead to hereditary spherocytosis. Spherocytosis में actually hemolysis होता है, और because there is no biconcave shape, the cells actually, the RBCs are spherical in shape. So that is all about the morphology of RBCs. I hope it was clear. See you in the next class. Allah Hafiz.